This little graduated cylinder right here, it's basically my life story, right? Completely kidding. Moving right along, let's talk about acid-base buffer systems. So in order to understand what an acid-base buffer system is, we need to understand what buffers really are and what they're going to be doing, what their goal is inside of a system. Realistically, what a buffer is going to be doing is we're trying to manage the system. We're trying to help maintain that perfect 7.4 pH, that perfect homeostasis section, right? So if we begin to become too acidic, we start to increase the amount of hydrogen ions in the concentration, or amount of hydrogen ion concentration. If we start to increase the amount of hydrogen ions present, we need to make sure that we can actually pull that level back to within a normal realm to keep us at that 7.4 pH. The opposite is also true. If we start to decrease the amount of if we start to decrease the concentration of hydrogen ions too much, buffers are actually going to serve to increase the hydrogen ion concentration so that we can maintain that perfect pH balance of homeostasis. Makes sense? So essentially what a buffer is doing is serving as a game manager, right? Like it's a point guard. It's trying to decide who's getting the ball when, and how we're facilitating the play. It's an acid-base buffer system. It's serving as the point guard. Generally, buffers are going to be a weak acid and a conjugate base. Super complex. If we dive too far into that, it's not worth it. Essentially, know that it's a partnership. So it's an acid and a base. So if we have increasing levels of acidity, the base is going to be taken over. If we have increasing levels of alkalinity or decreasing hydrogen ion concentration, then we can we can throw in the ball from the point guard. If that's a good basketball reference, I don't think so. We can pass the ball. There it is. Back towards making more hydrogen ions and get us back level with our pH. With that being said, just like sports, there's some point guards or there's some buffers that are going to be better than others. We're going to have a couple different types of buffer systems. Let's talk about the intracellular buffers. These are going to be our first line of defense. These are going to be what we use really within the muscle to try to offset some of the hydrogen ion accumulation that's going to be occurring with the onset of exercise and with the increasing duration or intensity of exercise. 4 major classes here. We're going to have bicarbonate, we're going to have phosphates, we're going to have cellular proteins, and then we're going to have histidine dipeptides, which is essentially 2 linked amino acids that are capable of buffering hydrogen ions. The major one of these histidine dipeptides is going to be carnosine. If you want more information on carnosine, we'll talk about it in sports nutrition. If you take sports nutrition with me, I'm sure Mrs. Lane talks about beta-alanine supplementation as well. This is essentially the primary vector or the primary mechanism by which beta-alanine is working, is we're going to be messing with our carnosine concentrations. Interestingly enough, side note, squirrel, right? Like the primary company, the major company that has all the research backing behind it for beta-alanine is actually called Carnosyn, Carnosyn. And that's going to be like really the only major scientifically backed beta-alanine supplement that's out there. Kind of interesting, right? News you can use. There's also going to be hydrogen ion transporters. These hydrogen ion transporters are a way that we're actually able to maintain the hydrogen ion balance by either putting them into the tissue, like into the muscle tissue or into the blood, or into the muscle tissue or into the blood. There's going to be a sodium hydrogen exchanger, or like an antiporter. So what these are going to do is they're going to import sodium into the muscle in exchange for a hydrogen ion. So we're able to buffer the hydrogen ion concentration inside the muscle by straight kicking that sucker out of there and bringing in some salt, right? Bringing in some sodium. So that's an antiporter. We're also going to have symporters. We're going to have something that as it exports hydrogen ion, it's also exporting another substance. And those are going to be our monocarboxylic acid transporters, or MCT transporters. For those of you nerds out there, these are going to be organoacid transporters. So this is also going to be not just how lactate and hydrogen make it out. This is going to be how ketone molecules make it in and out of the cell as well, along with a whole bunch of other things, right? So MCT transporters are highly used, and they're also a hydrogen ion transporter to serve as a buffer. Make sense? Cool. So if these are buffer systems that are lying within a muscle, what do you think the difference is between type 1 and type 2 muscle fibers and their ability to buffer changes in hydrogen ion concentrations? It might be a little bit different than you would think at first glance, but it makes sense whenever you get at it, right? Essentially, what research indicates is that type 2 muscle fibers are better at buffering hydrogen ion concentration changes than type 1 muscle fibers. So type 2 muscle fibers are able to handle or buffer changes in pH better than type 1 muscle fibers, right? Like it sounds counterintuitive, but if we think about it, it makes more sense. Type 2 muscle fibers are going to be predominantly glycolytic, and they're going to be producing high rates of muscle contraction. What do we know about high rates of muscle contraction in hydrogen ions? Well, we know one, that we're going to be producing hydrogen ions from ATP breakdown using ATPase. High levels of muscle contraction, high levels of ATPase activity, high levels of hydrogen ion production. The other thing that we know, if they're highly glycolytic, they're going to be producing a lot of lactate. Production of lactate also is accompanied with production of hydrogen ions. If we're trying to get rid of lactate, we're also, if you go back to the last slide, going to be trying to eliminate hydrogen ions as well. So whenever we're eliminating lactate from the system and sending it on the Cori cycle, we're going to be buffering out some of that hydrogen ion, right? So there's a lot of things going on here, right? So a type 2 muscle fiber has a greater ability to transport out lactate and greater ability to transport out hydrogen ions. But we also, with training, can increase the carnosine concentrations, and we can supplement to increase the carnosine concentrations as well. So we can actually, in our type 2 muscle fibers, kind of bolster our buffering abilities with training and handle the acid load a little bit better. What about getting into the blood? What about the extracellular buffers? Well, we have three predominant types here. We're going to have proteins, low quantity, not really going to be discussed. Hemoglobin, pretty big, right? We know we got a lot of hemoglobin, if you remember back to a couple weeks ago when we were talking about the blood, circulatory physiology. And then bicarbonate. This is going to be the overwhelming majority of our buffering system inside of our blood, inside of our extracellular system, inside of our systemic system. Hemoglobin is just used for transporting oxygen, right? Like, isn't that what we learned? Well, it's not just that, right? Like, it is going to be used as a buffer as well. When we make it across the arterioles and we get into the venules, we're going to have deoxyhemoglobin, right? We're going to have already offloaded the oxygen and we're going to have deoxyhemoglobin. What else is being produced and then released inside of the capillaries, inside of the muscle? What's going to be released? We're going to be releasing CO2 and we're going to be releasing acid, right? Because our active muscle is going to be taking on oxygen and then dumping out waste products of CO2 and acid. Good thing is that deoxyhemoglobin is actually a very good buffer. It's actually able to bind hydrogen ions as CO2 is entering into the fluid. As CO2 is leaving the muscle and CO2 is entering into the bloodstream, the deoxyhemoglobin that sits on that comprises hemoglobin after it makes it through the capillary beds is going to actually be able to bind hydrogen ions, right? Like, that's how much more perfect could it get? As we're releasing hydrogen, or as we're releasing oxygen, our hemoglobin is actually, or deoxyhemoglobin more specifically, is grabbing onto our waste products too. So these are serving as both good shuttles and bad shuttles, right? Like, we're trying to get the good stuff to our muscles and we're trying to take the bad stuff away from our muscles. It's a super important thing inside of our blood. Now let's talk about the big mama jama, right? Like, let's talk about the bicarbonate buffer system. This is going to be the overwhelming majority of buffering that's going to be occurring, and it's going to be the single most important extracellular buffer that our body has. We can actually, side note, because I think it's cool, we can actually exploit this system and we can actually bolster this system by adding sodium bicarbonate into our pre-workout cocktails. So there's actually research where people will ingest baking soda, sodium bicarbonate, before a workout, and they will actually be able to enhance their buffering capacity and they'll be able to perform significantly better with exercise. they're able to delay fatigue significantly by supplementing with sodium bicarbonate. Side note to a side note to a side note, right? Like Inception. It essentially, side note to a side note to a side note, it's Inception, right? Side effect for the side note is going to be, it's oftentimes accompanied with GI distress. So think poop in your pants, essentially. Not quite that bad, but it's gonna cause some gastrointestinal distress for sure. Essentially, the way that bicarbonate buffer system works is using these equations right here. So let's look at this second equation because this is more specific to the blood. The top one is how sodium bicarbonate works. Essentially, you have carbonic acid. So remember, as CO2 enters the blood, it combines with water, we talked about this, and creates carbonic acid. Carbonic acid is going to be a weak acid. We're also going to have a sodium hydroxide, which is going to be a weak base. So remember, our buffer systems are going to work on a acid and a conjugate base. Here you go. We're going to have a production with carbonic acid in the strong base sodium hydroxide. We're going to produce bicarbonate in water. How this is going to work is going to be found, I hope I'm pointing in the right direction, down here in this figure, right? So at the active tissue, we're going to produce CO2. We're going to release that CO2 from the cell into the bloodstream. Inside of the blood, we're going to have a combination of CO2 with H2O. I almost called it dihydrooxygen. Anyways, we're going to have a combination of carbon dioxide with water, and we're going to produce carbonic acid. Carbonic acid is going to get broken down into a bicarbonate molecule and a hydrogen ion. The hydrogen ion is going to be bound to deoxyhemoglobin, right? We just went over this on the last slide. We're going to release that bicarbonate, and that bicarbonate is going to be circulating inside the blood. When we get closer to the lungs, we're going to have basically a reversal of that reaction. We're going to have a bicarbonate. We're going to create carbonic acid. Carbonic acid is going to get broken down into CO2 and H2O. Release of the carbon dioxide. Carbon dioxide is going to make it into the lungs, and we're going to exhale. Right? Essentially, that's how the bicarbonate buffer system works. In the big picture, in the major thing that I want you guys to understand is that we're able to use bicarbonate to buffer CO2. And that CO2 can then be, that CO2, that bicarbonate can be transported throughout the blood, offsetting the acid load. And then when we get closer to the lungs, we can kind of flip the script and get rid of that and eliminate that CO2. This is the overwhelming majority way of how we handle an acid load inside of the body. Essentially, our bicarbonate to carbonic acid ratio is going to be about 20-1. So we're able to handle a high acid load. And it is a very effective system for getting rid of acid. So why does this reaction occur? Why does this go on? Well, essentially, this is going to be occurring because of respiration. Respiration is going to be driving this buffering system. If you go back a couple of slides, we talked about CO2 as a volatile acid. CO2 being a volatile acid, meaning it's a gas, and meaning we can exhale it. So the amount of CO2 that's in the blood is going to drive the carbonic acid production. So if we have high levels of CO2 inside the blood, we're going to drive the reaction to the right, and we're going to create high levels of carbonic acid, right? H2CO3. If we have high levels of H2CO3, we're going to produce high levels of bicarbonate. When we reach the lungs, we're able to essentially use carbonic anhydrase to drive this reaction in the opposite direction and eliminate CO2. When we eventually eliminate that CO2, we're able to rapidly drop the pH because we're able to rapidly get rid of this system. We're able to rapidly reduce the need to rely on this system and eliminate the acid load within our body, right? So the major take-home point, we have multiple buffer systems. We have buffer systems inside the muscle. We have buffer systems in the extracellular environment or in the circulation. We've talked about the ones inside the muscle, And the bicarbonate system is the overwhelming majority of buffering inside the blood. Essentially, what we're doing is we're using bicarbonate to buffer out the change in acid load inside the body. That bicarbonate helps facilitate the expiration of the CO2, which helps to drop our acid load, or it helps to maintain our acid load, or it helps to reduce our hydrogen ion concentration and thus improve our acidity or make us less acidic. Now, these are all, for the most part, short-term, more acute levels of handling or ways of handling acid load. There is another kind of more long-term control mechanism. It doesn't really play a huge role during exercise, at least short duration exercise. You got to be going on for several hours before this really starts to take effect. But essentially, the kidneys are actually able to manipulate the level of bicarbonate inside of the system. So as the pH drops, as we get more acidic, the kidneys are going to decrease the excretion of bicarbonate. So we're going to make sure that we are facilitating greater levels of buffering to get essentially eliminate the hydrogen ion concentration with bicarbonate. As the pH increases the opposite direction, the kidneys are actually able to excrete more bicarbonate and help bring the pH down back into a realm that we're wanting it. So kidney function, kidney level regulation of acid-base balance is more long-term, not directly applicable to short-term or more acute exercise physiology. So what's occurring with acid-base balance or acid-base regulation during exercise specifically? Well, the final stages of an incremental exercise test is really going to be where we start to see an increasing acid load or an increasing hydrogen ion accumulation. So a more acidic environment. We can also get this with high levels of anaerobic activity. Think repeated sprints. Think repeated 40-meter dashes or repeated 400s. You can force your body to become very acidic very fast. And it's largely going to be due to an increased concentration of hydrogen ions, right? We have those three mechanisms for how we're making hydrogen. It makes sense why these types of exercise activities are going to producing hydrogen ions in high levels. And what I want you to look over here is that muscle pH is going to be significantly lower than blood pH, right? We're able to handle and able to buffer blood pH significantly better than we are inside of the muscle because of that bicarbonate buffering system, right? Our muscle doesn't have that at all. So we're having to use the blood pH. So we're having to really use bicarbonate inside the blood to manage this. Trying to think about what type of exercise can produce the greatest level of acidity or decrease the pH the most. We're going to have three kind of primary regulators. We're going to have exercise intensity, we're going to have amount of muscle mass involved, and we're going to have duration of the exercise. All three of these make sense with kind of those three causes of the change in hydrogen ion concentration, right? Exercise intensity, as we become more intense with our exercise, we're going to become more glycolytic. If we're more glycolytic, we're going to be producing more lactate. If we're going through more muscular contractions, that could be exercise intensity or amount of muscle mass involved or duration of the exercise, we're going to be breaking down ATP at high levels. So we're going to be producing hydrogen ions, right? The other one, duration of exercise. If you think about duration of exercise means that we're using aerobic metabolism quite a bit more. That means we're going to be burning the Krebs cycle and the electron transport chain at full tilt or at full steam. So we're going to be breaking down pyruvate into acetyl-CoA pretty fast. So we're going to be liberating carbon dioxide pretty rapidly. So we're going to be producing acid. See how this all kind of ties back? So if we're trying to predict what exercise is going to produce the biggest acid load, well, exercise involving high intensity with large muscle masses. Think leg day, right? Think heavy, high rep squats. I've actually done 6x10 squats at 85%. Clearly I didn't survive. Well, I guess I did because I'm here, or am I here, right? So there's exercise programs that you could do that are basically brutal for your legs that are going to induce a very rapid shift in acid-base balance. And the lowest ever recorded is actually 6.8, right? Like, that's crazy low. That is crazy low. That can be achieved, well, close to that. I don't know if close to that's even appropriate. You can drop the pH pretty rapidly and pretty extreme with high levels of lower body exercise, especially resistance training exercise or repeated wing gaits or whatever. And that tends to recover back to a normal level. If it doesn't recover back to a normal level, go see a doctor because it's life-threatening. As we're making this acid, we have two lines of defense to really think about. Our first line of defense is going to be residing within our muscle, right? Because that's where we're producing it. We need to be able to buffer some of the hydrogen ions that we're producing. We're going to do that inside the muscle. So about 60% of that buffering capacity is going to come from intracellular proteins and histidine dipeptide or carnosine. 20 to 30% is going to come from muscle bicarbonate, and then 10% is going to come from intracellular phosphate groups, right? Just know that the bulk is going to be coming from intracellular proteins and histidine dipeptide or carnosine. Our second line of defense is going to be what's occurring inside of our blood. And that's that bicarbonate blood system, that's the deoxyhemoglobin, all that, right? At approximately 50 to 60% of VO2 max, that's when we're going to start to see an increase in blood levels of lactate. And we're also going to see a pH decline that's associated with this, not at the exact same point necessarily, but we're going to see a direct decrease in hydrogen, or we're going to see a corollary decrease in pH levels. So we're going to see an increase in hydrogen ion concentration. We can actually sense this in our carotid bodies, right? The same things that are serving as oxygen sensors and all that are going to be able to signal to the brain that, oh my God, we have an increasing acid load. We have decreasing pH that's occurring inside of our blood. We can sense it, we can feel it, we can see it, and we can send that signal to our brain. That medulla is then going to take and interpret that signal, and then we're going to return back and start exhaling at a higher rate. We're going to start manipulating our ability to expire air to start buffering off some of that CO2 or start increasing our pH or decreasing our hydrogen ion concentration. So in general, we're going to have two lines of defense. We're going to have a chemical buffer system. We're going to try to handle our stuff inside of the muscle, or let's go a little bit more global. We're going to try to handle our stuff inside of muscle and blood. And our second line of defense is going to be, how are we handling this physiologically, or how are we handling this on a bigger picture, on a global picture? And that's going to be with managing at a kidney level long term, or increasing our expiration inspiration so that we're able to manipulate the CO2 concentrations inside of the blood. I know it was a lot. Watch this, rewatch this, definitely be reading. Acid-base balance is really where all this starts to come together. So it's super important that you guys get this. Reach out to me. You know I'm always here. See you guys in class.